Dr. Gagandeep Kang is professor in the Department of Gastrointestinal Sciences at the Christian Medical College, Vellore, India, and was till recently the executive director of the Translational Health Science and Technology Institute, Faridabad, an autonomous institute of the Department of Biotechnology, Ministry of Science and Technology, Government of India. She is a leading researcher with a major research focus on viral infections in children and the testing of rotaviral vaccines. She also works on other enteric infections, sanitation, and water safety. She was awarded the prestigious Infosys Prize in Life Sciences in 2016 for her contributions to understanding the natural history of rotavirus and other infectious diseases. In 2019, she became the first Indian woman to be elected as a Royal Society Fellow. She played a significant role in the efforts that culminated in the development of Rotavac, a vaccine from Bharat Biotech International that targets diarrhea. She was one of the three principal investigators in the phase 3 clinical trials of the vaccine. We welcome Dr. Gagandeep for her talk on the immunological response and vaccination in COVID.
Hello, I'm Gagandeep Kang, and I'm going to be speaking to you today about immunological considerations for COVID-19 vaccination strategies. In order to understand what are the important immune responses, we need to know a little bit about the structure and the replication of coronaviruses. There are major structural proteins of the coronavirus. The first is the nucleocapsid protein, which is bound to the RNA genome. And then we have the spike protein, which forms the corona on the surface of the virus. This is critical for binding to the host cell receptors to facilitate entry of the virus into the host cell. Then we have the envelope protein, which interacts with the membrane protein to form the viral envelope. And the membrane protein determines the shape of the virus envelope and is the central organizer of coronavirus replication. When the virus gets inside cells, other proteins, which are called non-structural proteins, are also made, and it's possible to make an antibody response against these. So the antibody repertoire that is generated by a coronavirus infection can consist of responses to the structural proteins as well as the non-structural proteins. Among the non-structural proteins, there have been a few that have been identified as being very relevant, but the focus mostly has been on structural proteins, and particularly the spike protein, on which many of the vaccine constructs are based. When an infection happens before symptoms start in the pre-symptomatic phase, or if a person stays asymptomatic, you can start to see an antibody response that is generated several days after the virus becomes detectable. So if we look at RT-PCR, you can have RT-PCR positivity in the pre-symptomatic period, and then it rises to a peak after the onset of symptoms. When we look at what happens to antibody responses, those tend to be generated a few days after symptom onset. And unusually in this virus, unlike the standard pattern where we see IgM antibodies first, here IgM and IgG rise almost simultaneously, just a couple of days apart. And then IgM levels fall about 3 to 4 weeks after infection, while IgG levels, depending on what viral protein they are directed against, persist for a reasonable length of time. In addition to the antibody responses, we also have cellular responses that are made. And these cellular responses can be driven by many classes of T cells. In addition to the antibody acute response, there is a memory made by memory B cells, and assays for memory B cells and for memory T cells are very relevant to vaccine development and use, because it is through these assays we will learn for how long these vaccines will continue to work to provide protection, and whether or not boosters are going to be required. If we examine the assays that are needed, therefore, the assays are for antibodies, they are for memory T cells and for memory B cells. Among the assays for antibodies, we measure total binding antibodies. Those can be of different classes: IgG, IgM, IgA. We can look for antibodies to the spike protein, to the nucleocapsid, as well as to structural and non-structural proteins. We can look for functional antibodies, which are usually the correlates of protection for other viruses and maybe correlates of protection here as well. These are neutralizing antibodies that prevent the virus from entering inside the host cell and replicating. In addition to this, we can look for function of the Fc portion of the antibodies. If we look at antibodies as being Y-shaped structures, the stem of the Y is the Fc portion. And here we can look for non-neutralizing or extra-neutralizing functions. For memory T cells, we can measure the quantity of antigen-specific CD4 and CD8 T cells and also characterize their cytokine profile. For memory B cells, we usually measure the quantity of antigen-specific memory B cells in blood. All of these assays taken together give us a reasonably comprehensive picture of what is happening in infection and when a vaccine is given. Now, if assays for total binding antibodies are what we know as serology assays. These are the ELISAs. They can be done on different kinds of platforms. It can be just a regular plate ELISA. It can be modifications of enzyme immunoassays, such as chemiluminescence assays, which change what you use in order to detect a signal. Now, what are we measuring here? We are measuring the levels of total binding antibody or specific classes of antibodies against a variety of defined proteins. The ones that are important to remember so far are the spike and the nucleocapsid proteins. And why do we want to know this? Well, these antibodies are actually very easy to measure. But what we are really interested in is what proportion of these total antibodies are functional antibodies that will actually correlate with protection from infection. The epitopes, if we examine the structure of the virus, the spike is what is being considered most often. And in the spike, we are looking mostly at the receptor binding domain, which is the path that binds to the ACE2 receptor. How is the assay performed? The assay is performed by coating the antigen of interest on a solid phase. This can be the kinds of plates that we know as ELISA plates. It can be beads. It can be a variety of other substrates. And then the antibody that is present in the serum binds to the antigen, and there is a detection system. There are now new systems being developed that allow you to measure not just a single antibody to a single antigen, but to measure multiple antibodies to many antigens. If we examine neutralizing antibodies, these are functional assays. They look to see whether there is an antibody present that prevents the interaction between the spike protein and the ACE2 receptor. And it is again the spike and the receptor binding domain of the spike that are most relevant here. There are many ways of doing this assay. in the conventional viral neutralization assay, this needs to be done in a BSL-3 facility where you take the antibodies, you incubate them with the wild type virus, and then see whether the presence of antibodies inhibits the replication of the virus in a cell culture system. This is difficult to do because it requires a BSL-3 facility. So there have been substitutes that have been found. One is to make pseudoviruses. Instead of using wild type virus, you can just have some other virus which carries the spike protein and has it on its surface so that a pseudovirus neutralization can be carried out. And this can be done in a BSL-2 facility. So you don't need the same kind of containment. It still takes time to get it done and it can be hard to standardize, but it removes at least one level of biosafety requirement. In addition to that, another test has been developed, which is called a surrogate neutralization assay, where you just take the spike protein and the ACE2 protein and you look to see whether the binding of the spike protein to the ACE2 protein can be inhibited by the presence of antibodies. It's almost like an ELISA format and can be done quite quickly. In addition to that, there are also assays for the FC-mediated functions of antibodies, and this is really looking to see whether antibodies can lead to uh, actions of the immune system that result in the destruction of the virus or of virus infected cells. So in antibody dependent cell mediated cytotoxicity, what you see is that you have expression of the viral proteins on the surface of infected cells, and the FC portion of the antibodies that binds to the viral antigens on the surface can also interact with FC receptors that are present on natural killer cells. And this results in a destruction of the infected, virally infected cells. Now, this is relevant in natural infections. We don't know whether this will play a role in vaccination, but it is important for us to understand that there are multiple ways in which virally infected cells can be removed from the host. Why do we need these for vaccination? I think there is only a little bit that is being done in this area, but it will be important for SARS-CoV-2 vaccines because different vaccine platforms induce antibodies which have different effector functions. So some vaccines may drive immune responses that may be protective, whereas other vaccines may drive immune responses that may result in pathology. Antibody dependent enhancement is at least a theoretical concern. It has not yet been demonstrated in COVID-19, but we do need to keep a watch for that. Now, what are all the different kinds of platforms that are being used to make vaccines? We have live attenuated and inactivated vaccines that consist of the whole virus. We have virus-like particles. We have just the protein subunits. We also have vectored vaccines where the gene that encodes for the spike protein or for the receptor binding domain is carried by another virus into the host cell. We have DNA and RNA vaccines, which also encode for the spike protein and essentially use the host cell as factories to make the spike protein to which the immune system then responds. It's important to understand which are the proteins that are available to make an immune response so that in the future it will be possible for us to distinguish between vaccinated and infected individuals. So if you have a live attenuated or an inactivated vaccine, we may not be able to distinguish infection from vaccination. But for all of the others, where only the spike is being delivered, it may be feasible for us to say this person is vaccinated because they have antibodies only to the spike protein and not, let's say, to the nucleocapsid protein, which would be seen with a whole viral particle. Now, if we examine the vaccine platforms that are available and the kinds of antibody responses and cellular responses we expect to see, we are already seeing strong neutralizing antibody responses with all of the vaccine platforms that are being tested. And this is really good news for the development of vaccines. Where T cell memory responses are being measured in viral vectored vaccines, in live attenuated vaccines, and in DNA vaccines, we would expect to see a strong Th1 polarization, and this is what we would like to generate. In mRNA vaccines and in inactivated vaccines, in protein subunit and VLP vaccines, it's very important that we have the right kind of configuration of the vaccine, which can depend quite significantly on the adjuvant, to make sure that the response is tailored more towards the Th1 type and not towards Th2. For CD8 T cell memory responses, these are generated very well by live attenuated vaccines and by viral vectored vaccines. With other vaccine types, you don't necessarily see a strong response. What this will mean for vaccines that are made on these platforms is something that we will only know over time. I'd like to switch here to give you a little bit of data from one new vaccine, the first one that went into humans. This is the Moderna vaccine, which is an mRNA vaccine, and the first trial that was done tried 3 doses of the vaccine and 3 different doses given as 2 injections to 15 people each. After the first vaccination, anti-spike antibodies were very well seen. And after the second vaccination, there was an increase in binding antibodies and in neutralizing antibodies. So if we look at the data here, what Moderna did was to compare the antibodies that were produced by the 3 doses of vaccine with what was seen in people who had recovered from infection. And if we look at figure A here, this is the spike protein, and you can see that there are good antibody responses made by all 3 doses that were tried. In figure B is receptor binding domain, a portion of the spike, and again, very good antibody responses are being made, somewhat above those seen with the convalescent serum. A pseudovirus neutralization assay is seen in C, and here again, with all 3 doses of the vaccine, there is a good neutralizing antibody response that is made. Some dose response here with 25 micrograms giving lower neutralizing antibody titers. And then a wild type neutralization, a plaque reduction neutralization test is shown in figure D, and again, good neutralizing antibodies were made. Now, this was the immunogenicity data of the vaccine, and we now have data that shows us that this is a vaccine that is working very well to protect against disease and against severe disease. One of the first 2 vaccines to have their data available from their phase 3 efficacy trials. Now, with all of this information, what do we want to do to inform vaccination strategies? What do we want? We want robust, long duration protective immune responses with a single dose in all ages, including those with immunocompromise or with comorbidities. Have we got that yet? Not yet. We know protection, but we don't know how long this protection is going to last. So far, we have not achieved this with a single dose. The vaccines that we are using are two-dose vaccines, and we do not know whether this will work in people with immunocompromised or with comorbidities, though the data are encouraging in that the elderly appear to be protected. One of the questions that comes up about vaccination strategies is, should we think about not vaccinating those who have already been infected? And this is, of course, very challenging to do because it would require that we ask everybody to have an antibody test before we consider them eligible to receive a vaccine. And that has never been done before for any vaccination program. So the way I'm thinking about this right now is that vaccination actually provides a known dose, whereas for infection, the dose that causes the infection is unpredictable. So we don't know whether the immune response will be sufficient to induce protection from every case of natural infection or not. We don't also know whether pre-existing antibodies could potentially interfere with vaccine take. So in terms of learning more about the immune response in the context of prior exposure, a lot more has to be done, but we should immunize those with and without a history of prior infection until we learn more. Thank you very much for your attention.